Good morning, my name is Maria Luisa Mendez, and today I'm going to talk about an article of molecular biology. A myocardial infarction can be defined as a myocardial necrosis due to an acute obstruction of a coronary artery. Symptoms may include chest discomfort with or without dyspnea, nausea, and diaphoresis. It can be classified into 5 types and can be diagnosed by several things. CRISPR-Cas9 is a tool used for editing parts of the genome. It consists of 2 molecules, and scientists developed this tool by adapting a similar gene editing system that some bacteria use to respond to pathogens. CRISPR-Cas9 has a potential tool for treating medical conditions that have a genetic component. Then we're going to talk about the objectives of the study. The first one is to develop CRISPR-Cas9 magnetoplexes that magnetically guide CRISPR-Cas9 system to the heart for efficient in vivo therapeutic gene targeting during heart failures. Number 2 is to demonstrate that the in vivo gene targeting of MIR34A via these CRISPR-Cas9 magnetoplexes in a mouse model of myocardial infarction significantly improved cardiac repair and regeneration to facilitate improvement in cardiac function. The first method used was immunocytochemistry. This technique is usually used for the detection of proteins and antigens in cells. It uses a specific primary antibody to bind to it. It was used to identify the proliferation of Cas9 magnetoplex-treated cardiomyocytes post-H2O2 induction, which means post-stress induction, by the recognition of PH3, which basically marks the active phases of proliferation and alpha-actin expression. The second method used was RNA isolation and qRT-PCR. RNA isolation helps separating RNA from tissues, DNA, or proteins. It is based on liquid phase separation. Meanwhile, qRT-PCR enables detection of products generated during the PCR process. It is based on an oligonucleotide probe that hybridizes within the target sequence. Cleavage of the probe during PCR due to Taq polymerase, which was used in this study, can be used to detect amplification of the target specific product. This technique was used to isolate mRNA from cells, and then the structured mRNA was reverse transcripted. The purpose of using this method was to examine the efficiency of the Cas9 magnetoplexes mediated genome targeting. The next method we used was Western blot analysis. In this technique, a mixture of proteins is separated based on molecular weight by electrophoresis. These results are then transferred to a membrane, producing a band for each protein. The membrane is then incubated with label-specific antibodies to protein that we have interest on. It was used to analyze Notch1 and peanuts in cells treated with MIR34A Cas9 magnetoplexes. Also, it was used to identify caspase-3 and actin in cells treated with these magnetoplexes. The next method they used for this study was Sanger sequencing analysis. This method involves electrophoresis and is based on the random incorporation of chain-terminating deoxynucleotides by DNA polymerase, which during in vitro DNA replication. This method usually is used to study a small subset of genes linked to the defined phenotype, or confirm next-generation sequencing variants, or detect minor allele fractions, or read contagious sequences. In this study, it was used to examine whether Cas9 magnetoplexes can cleave the target MIR34A in vivo. For the next part, we have the results. For this, we can say that as the gel runs, shorter DNA fragments will travel faster through the pores of the gel matrix than larger fragments, which basically means that shorter DNA fragments will go further in the gel than larger fragments. This means that they introduce correctly the magnetoplexes to a CRISPR-Cas9 vector, because it means that there's more DNA. In this image, we can see that they are showing us the results of proteins. If the line is thicker, it means that it has more expression. The results show that the MIR34A, after the introduction of the CRISPR-Cas9, had less expression, and that was what they wanted, because the MIR34A can downregulate certain targets such as Notch1 and peanuts, which help with the cell survival, DNA repair, and cell growth. The results showed that the Notch1 and peanuts had more expression after the introduction of the CRISPR-Cas9 treatment, which is a good thing for the study, because the Notch1 and peanuts help with cell survival, DNA repair, and cell growth. In here, the caspase-3 has more expression when there is not an introduction of CRISPR-Cas9. For the next part, we have the results of an immunofluorescence, where the fluorochromes used are DAPI, CTNT, and alpha-actin. These fluorochromes are marking different regions at the cellular level. These results show greater expression of proteins in post-myocardial infarction heart treated with MIR34A Cas9 magnetoplexes. This is because the MIR34A expression is accompanied by the downregulation of several direct targets, including Notch1, SID1, P10, and peanuts, which means that these proteins, after eliminating the MIR34A, began to show and increase the cell survival, cell growth, and DNA repair. We can say that both premises were demonstrated in the study. For the last part, we have the conclusions. The first one is that molecular biology has managed to open doors that were previously considered impossible in the world of medicine. The second one is that molecular biology is essential nowadays because of the various techniques that have been created with it.